Welcome to The Explainer. Got an exam on gene regulation coming up? Well, you're in exactly the right place. Today, we're going to break down all the essentials of how our cells control their genes and turn it into a super high-yield study guide to help you absolutely ace this topic.
All right, let's just jump right in with the big question, the one that gets to the very heart of gene regulation. Think about it. Every single cell in your body, whether it's in your heart or your brain, has the exact same DNA blueprint. So how on earth do they end up so different? Why does one become a heart cell and another a neuron? Well, that is the puzzle that gene regulation solves.
And the answer really is all about control. Gene expression isn't just a simple switch that's on or off. It's more like a really sophisticated assembly line with checkpoints all along the way. You can see it in this diagram here. There are control points right at the DNA level, then during RNA processing, then out in the cytoplasm during translation, and even after the protein is made. But the main event, the biggest control point and our focus today, is that very first step, transcription.
So this is basically the cell's main command center. It's what decides which blueprints from that massive DNA library actually get pulled out and read. And you can imagine, getting this right is absolutely critical for a cell to do its job properly.
So who's in charge of making these decisions? We'll meet the transcription factors, or TFs. You can kind of think of them in two main flavors. First, you've got your general transcription factors. These are the bare minimum, the non-negotiable ignition key you need just to get the RNA polymerase engine to start. And then you have the regulatory transcription factors. These are the fun ones. They're the gas pedal and the brake that control how fast or slow that engine is running.
And here's where it gets really cool. Look how these regulatory factors fine-tune everything. On the left, you've got an activator protein. It binds to a special DNA sequence called an enhancer, and that just cranks up transcription. It's hitting the gas. Then, on the right, you see a repressor protein binding to a silencer sequence. That's a brake. It shuts things down. So the cell is in this constant state of balancing the gas and the brake. Now, here's a little detail that pops up on exams all the time, so you want to remember this. Take a look at that location on DNA column. Where do you find these enhancers and silencers? And get this, they can be almost anywhere. They can be thousands, even tens of thousands of base pairs away from the gene they control, upstream, downstream, or even hidden inside an intron. The DNA is flexible and can actually loop around to bring these distant sites right up close to the gene's starting line. It's pretty amazing.
Okay, so we've met the team. Now let's actually see them in action. We're going to walk through step by step the exact sequence of events for flipping a gene into the on position.
It all happens in this really coordinated four-step dance. First, the activator binds to the DNA. That leads to the recruitment of a general factor called TFIID. Then the mediator complex gets involved. And finally, that kicks off the start of elongation. So let's break that down, starting with step 1.
So first up, the activator acts like a kind of talent scout. Its main job is to find and grab a general transcription factor called TFIID and bring it over to the promoter region of the gene. You can think of TFIID as the one that plants the flag. It marks the spot and basically shouts to everyone else, hey, the party starts right here.
Next, the mediator complex enters the scene. Now, if TFIID planted the flag, the mediator is like the project manager. It's this big protein complex that physically connects the activator, which is way out on its enhancer, to the RNA polymerase that's sitting at the start line. Mediator then gives RNA polymerase a little chemical kick, it phosphorylates it, which is the green light to go, go, go.
But wait a minute, what controls the controllers? I mean, these transcription factors are powerful, so their activity has to be regulated too, right? Right, and this slide shows the three most common ways that happens. First, a small molecule, like a hormone, can bind to it. Second, it can team up with another protein to form a dimer. Or third, it can be chemically modified, like having a phosphate group tacked on, which changes its shape and function.
And here's a perfect case study of that first method. Just follow along on the slide. You've got a steroid hormone, glucocorticoid, just floating around. It can cruise right through the cell membrane, find its receptor, which is actually a transcription factor, and bind to it. This new power couple then travels into the nucleus and gets to work, turning on specific genes. It's a direct line of communication from the outside world right to the cell's DNA.
Okay, so far we've been talking about all these proteins that read the DNA. But what if the book itself is slammed shut and locked away? That brings us to our next huge layer of control, how the DNA is packaged. It's not just about what can read the DNA, but whether the DNA is even readable in the first place.
You know, a really great way to picture this is to think of your DNA as a massive library. Some sections, which we call euchromatin, are like the open, accessible shelves. The DNA is loosely packed, the books are easy to grab, and so those genes are transcriptionally active. But other sections, heterochromatin, that's like deep archival storage. The DNA is packed so tightly you can't get to it, making it transcriptionally silent.
And this isn't just a theory. We see this in action in the most amazing ways. In female mammals, for instance, there are 2 X chromosomes. So to prevent a double dose of all those genes in every single cell, one of those X chromosomes gets randomly shut down by being condensed into this tight little bundle called a bar body. That's a perfect visible example of heterochromatin. Another really powerful way to silence genes is through DNA methylation. This is where the cell adds a tiny chemical tag, a methyl group, directly onto the DNA. It's like putting a do not disturb sign on a specific gene. And this works in a couple of ways. It can physically get in the way and block activator proteins from binding, or it can recruit other proteins that help compact that DNA into silent heterochromatin.
But hold on, we're not done yet. Even after the RNA is made, that first draft, the cell still has more chances to regulate the final outcome before a working protein is ever produced.
Let's look at these really quick. With RNA processing, you have this incredible process called alternative splicing, where the cell can cut and paste the RNA in different ways to make multiple distinct proteins from just one gene. Then, at the translation stage, you have these tiny RNAs that can find a specific messenger RNA and just stop it from being read by the ribosome. And finally, even after a protein is fully made, it might still need to be switched on or off through post-translational modifications. It's just layers upon layers of control.
All right, that was a ton of information. So let's do a super quick flash review to make sure the most important high-yield stuff is really locked in for your exam. Ready? Here is your ultimate cheat sheet. Remember, activators boost, repressors block, TFIID initiates the whole process. Mediator kicks off elongation. HATs, those are histone acetyltransferases, they loosen chromatin to activate genes. And DNA methyltransferases add methyl groups to silence genes. If you can remember what these 6 do, you are in fantastic shape.
Okay, quick practice question. Feel free to pause and think. So a factor that increases transcription, that's an activator, and the distant DNA sequence it binds to, that's an enhancer. Simple as that.
All right, one more for you. What's associated with active genes? The answer is histone acetylation. Remember, adding those acetyl groups opens up the chromatin, making it active. DNA methylation does the opposite. That's for silencing genes. And we'll just end on this thought. All these mechanisms we've talked about, the chromatin changes, the methylation, this is the basis of epigenetics. It's this incredible field that shows how our environment, our choices, can actually leave marks on our DNA that change how our genes are expressed. Which leaves us with a pretty deep question. What does this mean not just for our own health, but for the health of generations to come? It's where biology gets really personal.